In this first part of Chapter 7 from our exercise physiology text, we're going to do a brief survey of the nervous system and focus on how the nervous system controls movements, the contraction of the skeletal muscles. We're covering a series of topics which you can use this as a guide to later on as you're studying the chapter. We won't go through this list of topics right now because they'll come upon us as we enter into the chapter. So the nervous system is an overarching control system of the body in which neurons communicate with one another and with the outside surface of the body, the deep organs of the body, and send commands out to control various organs, effectors they're called of the body. It also coordinates the endocrine system. We saw in the previous chapter that the hypothalamus controls the anterior and posterior pituitary gland, and hormones in the anterior pituitary gland control other endocrine glands and so forth. So two overarching control systems, one the nervous system lightning fast and the other the endocrine system a little bit slower in its mode of action. We'll talk about voluntary control of movement quite a bit, how the nervous system controls the skeletal muscles and at our behest, and also we'll talk briefly about reflexes, automated responses of the skeletal muscles to certain types of stimuli. So thinking of the nervous system as a whole, we should sort of break it down a little bit, and we can think of the central nervous system as the brain and spinal cord, which then must be attached to the rest of the body in some way, and so all the neurons that connect the central nervous system to the rest of the body is called the peripheral nervous system. So we can further break down the peripheral nervous system into divisions. These sensory divisions bring information in to the central nervous system. Those are called afferent divisions. Afferent means incoming, and efferent divisions are called motor divisions. They carry information out. So sensory information brings, sensory divisions bring information in. Motor divisions send commands out to control the body. Here's a cartoon schematic to kind of clarify things. Brain and spinal cord is the central nervous system, and all the neurons that connect it to the rest of the body comprise the peripheral nervous system. We can then break things down into sensory or afferent divisions and motor or efferent divisions. Then we can break each one of those down into conscious or voluntary divisions. Somatic means voluntary or conscious sensory divisions versus visceral means involuntary or things that we're unaware of. And motor divisions can be broken down into somatic or voluntary outputs or commands versus autonomic or involuntary or things we're unaware of taking place in the body, but yet motor neurons are controlling effector organs. And here's fleshing things out a little bit more. We're thinking again here, over here on the left is the afferent sensory divisions, and the somatic sensory divisions are include all the sensations that we are aware of, that we can perceive. So touch and pain and temperature changes and itch and sight and hearing and so forth. All those are somatic sensory experiences. Visceral sensation means all the neurons that are monitoring our organs, our internal organs, that we really don't have any idea about until things go very, very wrong, and then sometimes there'll be a kind of a crossing over of sensations in the visceral divisions over into the somatic divisions, and we begin to get an idea that something's wrong. About output, the somatic motor division specifically is the control of the skeletal muscles. Those are the voluntary commanded effector organs, and the autonomic motor divisions control the organs and glands. We'll talk a lot more about those towards the end of the chapter. All right, now what are we talking about? We're talking about a system, a tissue type involving communication by way of neurons. Nerve cells or neurons are the part and parcel of the nervous system as far as communication goes. So here's a cartoon schematic of a neuron. It has a cell body, or soma, which contains all the familiar organelles that all cells have. There's a nucleus there, some endoplasmic reticulum, some Golgi apparatus, some mitochondria. So all the familiar parts are there. It's a living, working cell that can synthesize proteins and so forth and transport things. But it has a bunch of long thread-like processes sticking out that kind of make it unique. And those long processes attached to the cell body are called dendrites. They're like the antennas. They're information gathering structures of the neuron. And then there's a long process attached to the cell body carrying information out. And the axon is the process that carries information out from the neuron to either another neuron or to an effector organ. The axon has terminals down here, axon terminals, which, as we'll see later, are the site of neurotransmitter storage and release in order to communicate with the next component of the circuit. On the axon, these sort of hazy-looking purple barrels represent wrappings, layers and layers of wrappings of phospholipid bilayer. In other words, it's like having layers and layers of extra cell membrane wrapped around and around the axon in little segments with gaps in between them. That's called the myelin sheath. And the myelin sheath is essential to the normal functioning of the nervous system. Having these wrappings on the surface of the axon speeds up the conduction of impulses or activity 100 times or so. So we need to have this myelin sheath on here to really zip those signals, whatever they may be, as we'll discover, down the axon to the terminals so that we can communicate with the next element in the chain. When that happens, the axon terminals, which form synapses or junctions with other neurons or effector organs, will release neurotransmitters out onto the targets. Since we're talking about the myelin sheath, we'll quickly mention a disease, an autoimmune disease in which the immune system is attacking the myelin sheath of the neurons of the nervous system. It destroys the myelin sheath and ultimately the axons, and as it progresses, the person with this disease becomes fatigued, experiences muscle weakness, loses motor control and coordination, loses balance, and starts dealing with various personality changes and mood disorders. It's all about not being able to properly send signals at the appropriate pace down the axon of billions of neurons in the nervous system. One interesting thing about MS and many diseases is that, in spite of the fact that there's no cure for MS, exercise actually improves the quality of life and slows the progression of the disease to some degree. At least it improves the functional capacity in spite of this progressive disease. So exercise is a wonderful thing to promote, and it's important to have professional practitioners who can work with various populations, in this case special populations that have health issues, to know how to teach them to exercise and what kind of goals to set and what kind of exercises to do to maximize their quality of life in the context of this disease. Synapses, again, are the junctions between axon terminals and effector organs or other neurons. So a synapse is the little gap between an axon terminal and, in this case, the dendrite of another neuron, the antenna. And so an impulse, a signal, will travel down the axon of a neuron to the terminals, and neurotransmitter will be released, which will bind to receptors on the dendrites of a postsynaptic neuron. And then that neuron may become excited and send a signal down its axon to the terminals. It's also possible a synapse could form between terminals and an effector organ, such as a muscle. So that's what's meant by synaptic transmission. A signal from one neuron will then kind of leap across to another neuron or to an effector organ by way of the release of chemical signals. Now, it turns out that neurons and muscle cells are excitable cells. to understand how signals, what kind of signals are transmitted along the axon of a neuron and across the membrane of a muscle cell, we need to talk about the properties of the membrane that make for this irritability, this responsiveness of the cell membrane to various stimuli. And once a stimulus is encountered and it triggers an impulse, the impulse will travel or spread or be transmitted along the axon. So our goal is going to be to understand what it is that makes the cell sensitive to stimuli, and then what is an impulse and how does it travel, how is it transmitted. Well, to begin with, these cells have an electric charge on their membrane. That's an interesting thing, isn't it? They're like little batteries. There's a net negative electric charge inside the membrane and a net positive charge outside, an imbalance of electric charges across the membrane. So if we took a little voltmeter and measured the electric charge on the membrane, we'd see that it registers somewhere between typically -70 and -90 millivolts. That's a significant amount of charge. What is it that makes that charge happen? And so let's go there. There are two things to understand to explain why there's a negative charge on a resting neuron or a resting muscle cell. The first thing is that there are large concentration differences for sodium and potassium across the cell membrane. Those represent driving forces for diffusion, and that's created by the sodium-potassium pump. The pump maintains these differences in concentration across the membrane. Secondly, the membrane is more permeable at rest to potassium than sodium. So we'll take a look at those two things. The sodium pumps creating gradients and the difference in permeability of the membrane to the ions. So what is a sodium-potassium pump? It's a membrane protein that uses ATP, it's in all the cells in your body, to pump potassium ions into the cell across the membrane and sodium ions out. That's all it does all day. Collects potassiums into the cell and kick out sodiums. So consequently, potassium ions accumulate. And there's a high concentration of potassium in the cell, and sodium concentration goes down, or kicking it out. And so there's a low sodium concentration inside the cell and a high concentration outside. Incidentally, those outside concentrations are controlled by the kidneys, not by the pumps, but the pumps affect the gradients, create gradients, differences. So those are potentials, driving forces for diffusion. At rest, the membrane of these cells, the membranes of these cells, are more permeable to potassium ions than sodium. Now, the permeability means the ease with which the ions can get across. If the membrane has a low permeability, it's very difficult or unlikely for them to cross. A high permeability means they're wide open and the ions can cross. So at rest, the permeability for both ions is very low, but for potassium, it's about 100 times higher than for sodium. And so what happens is some potassium ions leak out of the cell faster than sodium leaks in, and that leaves behind a net negative charge. If you think of a cell as starting out with equal numbers of plus and minus charges, and we let some positive charges leak out, that's going to leave behind an imbalance, a negative pool of charges. So that's what happens, and we wind up with a negative resting potential, it's called. Negative resting membrane potential. Here's just a schematic cartoon diagram of the sodium-potassium pump working. Every cycle that burns an ATP, or you see it being hydrolyzed into ADP, takes that energy and transports potassium ions into the cell against its concentration gradient. In other words, there's a lot of potassium in here already. We're still able to keep pumping potassium ions into the cell from outside. And in addition, even though there's hardly any sodiums left in here, the pump can still keep grabbing sodiums and transporting them out of the cell, even though, again, there's a lot more sodiums already out here. That's the active transport phenomenon. So that's a sodium-potassium pump, and it generates and maintains the gradients, the differences. And here they are. So imagine this little sphere represents a cell, and in the membrane of this cell is the sodium-potassium pump, and it creates these gradients that you see listed here. Sodium ions are concentrated outside, low concentration inside. Potassium ions have a high concentration inside because they're being pumped in and accumulating low concentration outside. So that's the function of the pumps, to create and maintain these gradients or differences. Now the membrane is also somewhat permeable to potassium. They're called leak channels that allow potassium ions to just trickle out. They're not pouring out, but they're trickling out of the membrane, and as positive charges trickle out of the membrane, they accumulate kind of on the outside, leaving behind a balance of negative charges. There you have it. So here's our cell at rest. It has a negative charge inside the membrane and a positive charge outside. Pumps are running, ions are kind of trickling out, sodium ions occasionally are trickling in, and we reach steady state. We have a cell whose membrane has this negative charge on it at rest, the resting membrane potential. And here's just a little sort of visual of taking a voltmeter, a device that measures charge differences. Like if you stick a voltmeter on a battery, you'll see a voltage. That just means the battery also has a charge difference that can drive electric currents. Well, here's a voltmeter being used to test the charge difference across a cell membrane. And when we set up such a voltmeter, we'll see that there's in fact a negative charge inside the cell at rest of around -70 to -90 millivolts. These arrows or the dotted lines represent concentration gradients. It's like having a big crowd outside the gates of a venue before a concert or a ball game, and the doors are locked still, and all of those people are crowded out there pushing on the door, and they can't get through. That's the kind of thing to think about with a concentration gradient. If we were to open the doors, those ions would rush into the cell, just like the crowd rushing into the venue to get a good seat courtside to see the game. And if we opened the door for potassium, potassium ions would rush out through that doorway because they're crowded on the inside and they're pushing their way out. So that's the way to think about those concentration gradients. They can drive the movement or diffusion of ions. So that's our resting conditions. What about excitation? What about the impulses we were talking about? Well, to talk about those, we need to realize that on the membrane of these cells are some channels, some membrane proteins that can act as pores or passageways for ions to diffuse, and they can close and they can open so that ions can travel through. That's called gating. And there's a couple of kinds of channels to think about. Some channels are gated by chemicals that bind, neurotransmitter released from an axon terminal. If a neurotransmitter bound to a certain type of a channel, it would pop open like this, and ions would flow across the membrane. Other channels are responsive to the electrical properties of the membrane. Whenever the membrane is at rest and has a negative charge inside, the channel is closed. But if we let a bunch of positive charges come in and flip the voltage on the membrane, turn it more positive inside, then the channel would open and let ions cross. So, what's the stimulus? The neurotransmitter stimulates this charged membrane initially. The neurotransmitter is released from an axon terminal, diffuses across a synapse, and binds to its receptor channels, and that allows sodium ions to enter into that region where the terminal is, where the axon terminal is. If you have a net negative charge in the cell and you let some positively charged sodium ions in there, now you don't have such a negative interior. That's called depolarization. And if you depolarize the membrane enough, then some electrically gated channels are going to pop open, because remember, they're closed when the cell's at rest. But if we don't have that negative charge inside, those electrically gated channels open up, and we're going to have something that have an impulse, we call it. So that's called an action potential. So first, the neurotransmitter binds to its receptor channel, lets some sodiums in, that's the initial depolarization. Once that happens, some electrically gated sodium channels open because of that change in polarity, change in charge, and that allows a whole bunch of sodium ions to rush in at that location. Then a neighboring sodium channel, electrically gated sodium channel, pops open and more sodium comes in, which depolarizes that region. And then the neighboring sodium channel opens and it spreads across the membrane like a grass fire in California. Once you once you trigger this impulse or action potential, it'll spread across the entire membrane to the very axon terminals or across an entire muscle cell membrane, and nothing can stop it. Well, before long, the sodium channels close. They only stay open for a few milliseconds, and around that time, potassium channels start to open. They're electrically gated channels, just like these sodium channels, and they respond to the initial depolarization, but they just take longer. So a wave of depolarization spreads down the membrane, and then the potassium channels start to open and a wave of repolarization spreads. Why repolarization? Because remember, the potassium ions are more concentrated inside the cell, and so when we open up potassium channels, positive charges diffuse out, leaving a negative interior once again. So we have a wave of repolarization, reestablishing the negative charge in the cell. And then we can do it all over again. If there's still some neurotransmitter bound to its receptors, another impulse or action potential will spread down the axon to the axon terminal. Once it starts, you can't stop it. That's what's meant by the all or none law or principle. Here's another way of kind of looking at the same events to understand them. Imagine sticking the wires of our little voltmeter we had in the previous cartoon into the cell and outside the cell and measuring that resting membrane potential of -70 millivolts. And we just keep those wires in that set location, and then somewhere away from that spot we have that initial depolarization. We let some neurotransmitter come along, and then as the neurotransmitter generates an impulse and it spreads across the membrane, it's going to pass by the location where our voltmeter is situated, and we'll see some changes in the polarity or the potential on the membrane. So that's what we're looking at here. We're watching the voltage change on the membrane as the impulse sweeps by where we're watching. So initially we're reading -70 millivolts because that's the resting potential, and then we have this initial depolarization, and then it triggers an electrically gated channel to open, and as that happens, a ton of sodium can rush into the cell, depolarizing the membrane. Right? If it's very negative and we let in positive ions, the membrane is going to get more positive, and in fact it may reach as positive as +30 millivolts. Then the sodium channels inactivate, they stop letting sodiums in, and right about that time potassium channels begin to open, and a wave of repolarization passes by our observation point, and we see the membrane repolarizing as potassium ions are allowed to leak out and flow out through potassium channels, and we go back to our resting membrane potential. This word threshold refers to a certain amount of initial depolarization that's required before you trigger an electrically gated sodium channel to open. If we only had a tiny bit of initial depolarization, it wouldn't be enough and nothing would happen. But if we have enough neurotransmitter bound to enough receptor channels and enough sodiums rush in, then that'll depolarize the membrane to this so-called threshold value around -50 or 55 or 60 millivolts, in this case around -65 millivolts. Once we reach that depolarization level, then the electrically gated channels pop open and we have the action potential. So that's just another way of sort of picking up on what's going on during an action potential as it sweeps down the axon. So that's a little abstract, but it's a good thing to get a handle on, because now we understand what is an impulse, how does it travel down the membrane of a neuron axon to its terminals, and how does it travel across a muscle cell membrane. That'll be the subject of our next chapter. And now we can go on and understand more aspects of control of muscle contraction by the nervous system. So go back and review that a little bit, and it'll become more familiar. And join me for the next part of our story as we begin to take a look at the activation of muscles and other functions of the nervous system.